The overall goal of this recombinant HBV reporter system is to make it easy to conduct mass screening for anti-Hepatitis B virus or HBV agents.
This method can help answer key questions in the HBV field, such as screening for anti-HBV agents and identification of HBV host factors. The main advantage of this technique is that the HBV reporter system can monitor the early stages of the HBV replication cycle.
After preparing cell culture medium, the day before transvection, place 4 × 10⁶ Hep G2 cells in medium on a 10 cm collagen-coated dish. Incubate the cells at 37 degrees C in a humidified 5% CO2 incubator. Using a transvection reagent according to the manufacturer's instructions, transfect the cells with 5 micrograms of PUC1 2-HBV delta epsilon and 5 micrograms of PUC1 2-HBV NL. The next day, remove the culture medium and add 10 milliliters of fresh culture medium. Then, 1 week after transvection, transfer the culture medium containing the recombinant HBV to a 50 milliliter tube. Next, add 10 milliliters of fresh culture medium to the plate containing transvected cells. Then collect and centrifuge the culture medium at 2300 × G for 5 minutes.